Hey guys, how's it going? Hey guys, how's it going? Reed here. Today we're going to have a very interesting discussion on gene expression, specifically, the regulatory complexes that's responsible for gene expression. So, for example, when we have a gene, we need to regulate when it's expressed or not. And one of the ways that genes are expressed is you get the base pairs exposed. In order to expose the base pairs, you have to get all modifications of different type of proteins that change their 3D configuration. And when they change their 3D configuration, they don't the bundles don't align up properly, and so you get base pair exposure, and therefore you get continuous expression of those genes. So we're going to talk about regulatory complexes today, and we're going to see how they are a very integral part of regulating gene expressions. And this is important to know when you're prescribing medications, right? Because the medications that target G protein structures typically have instructions they want to send to the cell bodies to produce more of what that neurotransmitter is responsible for, right? And this is the process when the receptor binds to the G protein, it puts in a request to the cell body to make additional products. And this is the regulatory process that helps everything take place. Right. So let me see here. I look here, just Google explain regulatory complexes in gene expression. Regulatory complexes in gene expressions are intricate assemblies of DNA, right? RNA, protein, and protein molecules. Okay, you have the DNA. Here are the ingredients: the DNA, the RNA, and then all the proteins that make this happen. All right, they act together to control which genes are turned on or off, and when, right? Inside of a cell. These complexes coordinate the timing, location, and quantity of protein synthesis by interacting with DNA regulatory regions. So actually, for genes, you can have regulatory regions outside of the genes, also inside of the genes. So for example, like enhancers and promoters, and influencing the machinery, such as RNA polymerase, which is responsible for transcription. Let me read that over. They clump this all in one sentence. These complexes coordinate the timing, location, and quantity of protein synthesis by interacting with DNA regulatory regions, and influencing the machinery responsible for transcription, right? The resulting gene regulatory networks form a complex system that allows cells to respond to internal and external signaling, leading to specialized cell functioning, development, and the appropriate amount of proteins needed at a given time. Yeah, this is very interesting. Look at here. Okay, great. So it's the regulatory network. Wow. So the regulatory network forms the complex system that allows cells to respond to internal and external signals. Remember, I said when the neurotransmitter binds to that G protein is going to go through a 3D modificational change, and it's going to activate that protein kinase, the phosphorylase kinase system, and then we have that signal transduction pathway that creates a messenger RNA that sends the message to the nucleus. And this is the process where all of this timing and all. Now we have after you have the messenger RNA from that neurotransmitter that bound there, that RNA is going to be the template to create a protein that you're going to push out to the cell. So here, this is the regulatory. So I didn't put together that this is also the complex that forms here is also the structure that's responsible for specialization in cell functioning. And that would be really interesting, because if you have certain type of regulatory complexes, a high I guess, percentage of these, then that cell will specialize in this. I guess if it's other combinations, I guess it's the combination of the um, receptor complexes you have can lead to specialization. Didn't realize that's interesting. Development and the appropriate amount of proteins needed at a given time. Now, um, when you see how complex this dance is with all of these pieces, and it has to all take place in order for you to function, right? You can see how in developmental disorders, if the structure of the brain doesn't develop normally, how is it going to perform at this intricate level, at the molecular level? If the whole tissue is damaged or distorted, then how is this dance going to take place at the molecular level? I think you're going to have challenges here. I think things are going to be different there. You know, let's see here. Components of regulatory complexes. DNA regulatory sequences. And so these are sequences on the DNA. These are specific regions on the DNA that serve as binding sites for regulatory proteins, including promoters, enhancers, and operators. Great. Regulatory proteins. Transcription factors are a major type of regulatory protein. Transcription factors, that makes sense? That binds the DNA sequences, acting as activators and repressors. So the regulatory proteins can activate or repress, meaning promote gene expression or inhibitory. That makes sense. So we have the DNA regulatory sequencings. They serve as the binding sites for the regulatory proteins. The regulatory proteins are transcriptors. Transcription factors are a type of regulatory protein, and they can serve as activators and repressors. Mediator complex. In eukaryotes, the mediator complex acts as a scaffold, relaying signals from transcription factors to RNA polymerase to regulate transcription initiation and elongation. Right. So this mediator complex, it relays the signals from the transcription factors to RNA polymerase to regulate transcription initiation and elongation. So I guess a mediator is one thing that this mediator complex activates this RNA polymerase that then starts the process. Non-coding RNAs. These RNA molecules can also influence gene expression by interacting with DNA, proteins, or other RNA with these regulatory complexes. Histone modifiers and remodelers. Well, okay, this is the first time I heard that term. Histone modifiers and remodelers. In eukaryotes, chromatin structures must be open for gene expression to occur. Makes sense. Histone modifiers alter histone proteins, and nucleosome remodeling complexes can reposition nucleosome, making the DNA accessible to only transcription machinery. Let me explain here. Remember when I was describing that the DNA is sort of wrapped up around the histone, sort of like a spool of thread. Then there's a piece, then there's another thread, and there's a piece, and there's another, and then the whole thing sort of like comes on itself and things like that. All right, if you get phosphorylation of one of those proteins in that histone complex, that the presence of just one additional phosphate group or changes the entire 3D configuration of the molecule, right? And so if the whole 3D configuration of the molecule changes, then the DNA does not wrap around the histone like the spooling thread sort of model. Parts of it leaves sections of the DNA exposed, and now the transcription factors can get access to those base pairs and continue to transcribe and produce the product of what it's responsible for. But we see this in chronic cocaine dependency issues, right? In chronic cocaine dependency and use, you see a downregulation of the presynaptic dopamine receptors, right? And you also get a decrease in the a number of vesicles in the presynaptic receptor that contains dopamine. Therefore, with chronic cocaine dependency issues, you can become trending downward in more like a dopamine hypodopaminergic state because the system is just burned out, right? And the reason why that becomes permanent is because you get changes in the DNA. You get changes, not the changes in the DNA, you get changes to the proteins. And because the proteins are not allowing the DNA to be hidden and tucked away, sections of the DNA are chronically expressed. And that leads to the type of movement disorders, the type of behaviors and things that if it affects the motor area, it can cause other type of problems that's characteristic of these type of issues, right? So this is the important of this section here, this histone modifiers. That makes sense. This also happens in alcohol dependency also. You can sort of start to see a person with chronic alcohol dependency issue, particularly when they start to pick up the symptoms that never go away, right? Then you start to see the impact that substance is having on them. And that's happening at this level of modifying the histones and things like that. Okay, how regulatory complexes work. First, binding to regulatory DNA. Proteins bind to specific sequences of DNA, such as promoters or enhancers, right? You have to bind to the section that you want to express. Number two, interaction with other proteins. The bound proteins then interact with other regulatory proteins, forming large regulatory complexes. Okay, they start to assemble all of the pieces. Step three, influencing transcription machinery. These complexes influence the activity of RNA polymerase and other proteins involved in transcription, controlling the rate at which the gene is transcribed into mRNA. Make sense? Okay. Next, number four, responding to signaling. The entire regulatory complex can be activated or inactivated by signals from outside or inside the cell, such as hormone, nutrients, allowing the cell to adapt to its environment. This makes sense. It responds to it responds to influence from outside the environment. That's a very reliable system. You know, if you're going to produce things, I guess this is an input. Forming gene regulatory networks. Collections of these complexes, acting in concert with DNA and RNA, form complex networks that generate specific patterns of gene expression throughout development and in response to varying conditioning. That makes sense. Okay, let me see. Examples of regulatory complex. Lac operon in bacteria. So, you know, I don't think it's related to us. You know, I think that's all I wanted to say on this subject. Hey guys, how's it going? I appreciate it. Hey Patel. I think that's all I wanted to say for now. The 13 minutes mark, that's fine. If you have any questions, you can leave them in the comment section, and please like, subscribe, and share. Read out. Take care, guys. Ciao.